Happy Friday, North Texas! I'm Scott, and today is Friday, April 10th. Today, we'll be learning more about UNT's BioDiscovery Institute celebrating its 10-year anniversary. But first, we've got some news to know for our UNT community. Register now for the April 21st LinkedIn Open to Work Lab, an interactive experience where students will hear from industry leaders on how to navigate what work looks like now and what it means for you. Whether you're exploring career paths or getting ready to graduate, this experience is designed to help you take your next step with confidence. All attendees will receive a free copy of the book Open to Work: How to Get Ahead in the Age of AI. Lunch and career starter kit giveaways will be provided at the 11:30 a.m. session. Check the show notes for the event agenda and to register now. LinkedIn also is sponsoring a pop-up event from 9 a.m. to 5 p.m. Monday, April 20th through Wednesday, April 22nd on the first floor of the Business Leadership Building. All students are invited to attend regardless of whether they attended the Open to Work Lab. This event is designed to assist students in building their careers by helping them create or update their LinkedIn account, get professional headshots, learn how to stand out, and more. Join the UNT Alumni Association for Wine Down Wednesday, April 22nd at Four Corners Brewing Company in Dallas. Wine Down Wednesdays are monthly events designed to bring together our UNT alumni community from across DFW. Each event highlights a different college, school, or unit and the innovative ways they are shaping UNT's future. April's event will highlight UNT at Frisco. Check the link in the show notes to learn more and register. We've got exciting news to share about a fantastic opportunity for UNT undergraduate students this summer. The UNT Global Career Accelerator will be offered as part of a 6-credit-hour UNT course during the 10-week summer session, May 18th through July 24th, 2026. In this fully online, project-based program, you'll complete work connected to organizations with global reach such as Intel, OpenAI, L'Oreal, and the Grammys. Students will gain experience using industry tools including Google Analytics, Meta Ads Manager, Python, SQL, and Tableau, while strengthening the communication and professional skills employers expect. Learn more at careeraccelerator.unt.edu. And now for some athletics news, here's Zach Babb with the Mean Green Minute.
Mean Green softball dropped to 22-18 on the season following a midweek loss to Tarleton State in Stephenville on Wednesday. It was a 6-1 final for the Mean Green. They've now lost back-to-back games that followed up a 17-7 loss to Florida Atlantic last Friday. They now return home for a 3-game conference series. They're taking on East Carolina at Lovelace Stadium. The first game of the 3-game set will get underway on Friday at 6 p.m. Saturday's matchup is at 2 p.m. and followed by Sunday's series concluder at noon. All games are at Lovelace Stadium. And then they'll hit the road once more to Abilene Christian next Tuesday, April 14th. That's in Abilene, a 6 p.m. start there.
Thanks for that update, Zach. Today's interview is brought to us by Jacob Parker, a student assistant for UNT's BioDiscovery Institute. The BioDiscovery Institute was established in 2016 as one of 4 new institutes of research excellence. The institute's mission is to combine expertise in life sciences, engineering, and data science to discover and innovate next-generation biotechnologies, while training future scientists to solve health, food, and energy challenges. Jacob sat down with UNT Regents Professor of Biochemistry and previous director of the BioDiscovery Institute, Kent Chapman, to discuss how BDI has grown over the last 10 years and what the future holds.
I am just retired from faculty at UNT. I joined UNT in 1993. So have been in the Department of Biological Sciences for 33 years. About 2016, we formed the BioDiscovery Institute. Rick Dixon was the founding director. And from that time until, currently, I'm still a member of the BioDiscovery Institute.
Yeah, so it's to my understanding that it is BDI's 10th anniversary this year. What have you accomplished in the BDI since its inception?
Well, I think what I'm most proud about is the people, the talented people that we've been able to recruit to the Institute and to UNT. We started the Institute with existing faculty in several departments at UNT, and we made 5 key hires in the first 3 years of the formation of the Institute. And those people have joined, added expertise and knowledge and talents to the Institute.
What kind of talents? Like what do they specialize in?
Yes, that's a good question. We've added expertise in microbiology. So Dr. Hennard brought expertise in microbiology. Liz Skellum also in microbial systems and biosynthetic gene clusters and natural products in fungi. Serdar Bozdag brought expertise in computational life sciences and has started a center of his own and is a key member supporting our work in BDI. Ana Alonso joined BDI from Ohio State and brought with her a core facility in metabolomics, and that added expertise into the Institute. And Mauricio Antunes joined the Institute in that early recruiting period and has brought expertise in synthetic biology to the Institute.
Your specialization at UNT is with lipid research and metabolic processing and engineering, correct?
That's correct. I've been trained mostly as a lipid biochemist and cell biologist, plant cell biologist, and my lab has continued in those fundamental areas, although I must confess the technology has developed dramatically from when I first earned my graduate degree and when I first started my lab. So the things that we do are more sophisticated, more detailed than what I did when we first joined, but still focused on lipid biochemistry, lipid metabolism, and cell biology of lipids.
So with all of those things that you've done with lipids, of course, I am not the most scientific person myself. What can you tell me about what you've done and your work with them?
There's really a couple of areas where we've made contributions. One is in lipid signaling, and which we have worked for a number of years on a lipid signaling pathway that shares in plants, that shares similarity with a lipid signaling pathway that operates in humans. That's the endocannabinoid signaling pathway, and in plants, this signaling pathway seems to regulate responses of plants to stress and regulates growth and development. And more recently, I'd say in the last 15 years or so, we turned our attention to not how lipids serve as signals, but how lipids are actually produced and stored in plant cells. And that's been a fun project. Lipids are stored in plant cells like they are in most cells in organelles called lipid droplets. And we've spent the last 15 years or so trying to understand or identify the machinery involved at the cellular level in packaging lipids in plant cells.
So I actually did speak with another plant lipid specialist researcher, Peyton Whitehead.
Yes, PhD student.
Yeah, he was. Okay. He and I believe you contributed to an academic article that was in the plant cell.
That's right.
So can you tell me more about that?
Sure. That was an article that Peyton was first author. There were a number of contributing collaborators from, we had folks involved from Canada, from Michigan State, and from the USDA that were involved in this project. It was a project looking at how plants store waxes, how they produce and store waxes efficiently inside of lipid droplets. And Peyton discovered a protein that was very important in this process, and the paper describes evidence that supports the role of that protein in packaging waxes in plant cells. It was actually work that was started by another PhD student before Peyton, Drew Sturdivant, who did his PhD work with me as well, helped train Peyton, and some of the exciting results that Drew found are what Peyton carried forward.
So with Peyton and Drew, they were both students of yours at UNT. That's really interesting and exciting stuff to hear. But I'd like to know what drew you to UNT and the BDI.
Yeah, that's a good question. When I first came to UNT, I was a postdoc at LSU, and that's a temporary job. It's sort of like a residency after medical school. And so I was looking for a place to practice my science, and University of North Texas had an opening for a biochemist. And so I visited. I had not heard about UNT before, but when I came, I was very impressed with the location, with the people that were here, and with the resources that I would be able to use to get started and establish my lab. So I was drawn to UNT because of the emerging focus on research. When I came and when I actually joined, I was one of two plant biologists that were here. And so over the years, I've been very happy to have a dozen or so plant biologists join the faculty and provide great interactions as colleagues.
And the other plant scientist that was here with you, that was Rick Dixon?
No, the person who was here when I came, it was a plant biologist. He was a taxonomist. His name was Don Smith, Dr. Don Smith. He retired quite some years ago from faculty, but there just was not a focus in plant science here at UNT at the time. We did have a division of biochemistry. We did have a graduate program in biochemistry, and so I joined that program. And the plant scientists that then joined UNT primarily were in the area of biochemistry and molecular biology.
Okay, and you did have your own lab for a while, the Chapman lab.
That's right.
I do want to know more about that, about what it was like leading the lab and all the students that were in the lab, what the lab work is like at UNT. All that, but I will just start with the first question. What was it like leading the lab?
Well, it's one of the greatest things it's what I love about my job, really, is having a research lab, investigating questions. What drew me to science originally was curiosity and wanting to understand how things work in the living world, and being able to explore that is just a tremendous privilege. The best thing about it, though, is that I don't do it by myself. It relies on contributions from talented graduate students and undergraduate students and postdocs that have worked in the lab over the years. That's sort of a unique thing. You train, they learn the process of science while they're earning their degrees or their advanced training, and it gives me an opportunity to learn from them. There's nothing I enjoy more than sitting down with my students or my postdocs and going over their most recent research results. That's a fun thing for me. So I really, you might not think of it as leading a lab as much as coordinating laboratory activities. They all eventually work very independently, so they are deciding for themselves what their next experiment will be, what their question is that they're asking. And yes, we talk about it and we coordinate those activities around funded projects because there's no budget for research here at UNT, though that budget is something that comes in external federal sources of funding to the university.
And for those listening in who are students or undergrads or postgrads at UNT, they might want to know, how would you be able to join a lab for faculty?
Yeah, that's a good question. I think the first thing I would do is to look over the website, the information that's available on the BDI website, and see what people work on, and if there's anything that really strikes your fancy or, something that you might be passionate about. And then I would make contact with those research faculty, ask them if they have openings in their lab. If they are interested in training graduate students, or if they have openings for a postdoc, if that's the stage that you are in. And they would help guide you through the application process, probably starting a conversation and then an application to the graduate school and to the biology department. As a grad student, we have a rotation program in BDI. It's very new. Used to be that a student would need to have an advisor when they came in, couldn't be admitted unless they were already committed to working with an advisor. But now we have a rotation program where students can still work with somebody specifically, or they can come in and they can rotate through 3 laboratories and then make up their mind after they've rotated with those faculty on which lab they'd like to go into. That's a nice opportunity for the student to learn a variety of different techniques, but also to learn how the lab operates, the dynamic within the lab. Every principal investigator or lab leader functions a little bit differently in their level of expectations and direction that they give. And so it gives the student and the faculty member an opportunity to try each other out for a while before making a commitment.
And what kind of research projects in the labs or in the Chapman lab are you most proud of being a part of?
Oh, that's a hard one because everything we've done, I've been very, very proud of. I will make a couple of statements that really advances in technology have let us ask questions that we couldn't ask before the development of those techniques. And I'll give you an example of something that I still am fascinated by, and it's a technique called lipid imaging or mass spec imaging. It uses a mass spectrometer to map out the lipid metabolites within a cell, which is just absolutely fascinating. Instead of using a microscope to visualize components of cells, you use a mass spectrometer to identify the molecules that are present within the cell and then build back a 2-dimensional map of the distribution of lipids in a cell or tissue.
Is that like an MRI scan, like they scan like images of your brain and then you combine it together to make a full image?
It's similar to that. You could think of it as that because you are scanning across. In this case, it's a destructive sampling because you have to section the tissues, and then yes, you scan those tissues, but you're scanning for a signal. You're scanning for an actual signature of a molecule, similar as you would do with MRI. Yeah.
Okay. And what other technology or like technological feats have happened since you joined the BDI to now that have really helped with your lab work
besides the mass imaging? Yeah, that was a major advance because we learned that metabolism in tissues is not uniform throughout the tissue. We learned that in fact it's quite heterogeneous. There are places in which different metabolic pathways operate, and we were able to see that for the first time with this mass spectrometry imaging. Let me think of something else that has recently taken over the protein structure research area is called cryo-electron microscopy. It's not a new technique per se. It's a new technique applied to protein structure and function. And it involves very expensive electron microscopes that are able to get atomic level resolution, electron distribution of electrons around atoms of a protein, and then in a similar way take many, 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 many pictures of those protein particles and build back the structure, the 3-dimensional structure of a protein. And that cryo-electron microscopy, or cryo-EM, is a technique. It's a newer, advanced structural biology technique that has really revolutionized protein structure and function.
I see. So that's what EM stands for when I was looking at
electron microscopy.
Yeah, I was like,
yes.
What does that mean? So what kind of challenges did you have to face during your research?
Well, there are challenges at many levels when you're establishing a lab and maintaining a lab. And the first challenge is recruiting people. I mentioned at the very beginning that this is not something that is done by me in isolation. It's a team.
It's a collaboration.
It's a team, and building a team can be a challenge. Working together with others can be a challenge. Finding folks that are interested in the same things that I'm interested in is one level of a challenge. But once you find people and generate a team, the other challenge is, how do you pay for that team? How do you fund that team? How do you pay their salaries? How do you fund their research activities? How can you pay for their supplies so that they can ask their questions? That's a financial challenge.
Yeah.
So you've got a human resources challenge, you've got financial challenges, and then, of course, you've got scientific challenges. There may be many questions that you'd like to ask, but you're limited by equipment that's available, approaches that are available, know-how. And so one way to solve that, or one way to work around that, is to work with other people. So not just your team, but multiple teams working together, each with their own level of expertise or their areas of expertise. You can address so many more things than one team could do by itself.
Yeah.
So I'd say that those are challenges in science, and none of which are insurmountable.
Yeah.
Right.
And when it comes to the lab work and what you've done at the BDI and the research field, how do you view it all going forward? Like in the future, how do you view everything to be unfolding or progressing?
Yeah, this is an interesting time for BDI. There have been some retirements of senior faculty who were integral members of BDI. Dr. Dixon was the founding director of BDI, and he retired a couple of years ago. I have just retired. Rebecca Dickstein retired several years ago as well. So all of those people were experts in plant biochemistry. New people have joined BDI that were already here at UNT in other departments like biomedical engineering, material science and engineering, chemistry. And so the composition of members in the Institute has changed. It's changed in the last 5 years, but I see it changing even more and evolving even more going forward. So I'm excited to see how that will happen. Dr. Ana Alonso is currently the director in the Institute, and I noticed that this semester there were applications from a number of different researchers across the campus that would like to join the Institute. And if they do join the Institute, they would bring more, let's say, more on the medical side, more on the biomedical, biotechnology side of things. And so the Institute may evolve to have a different flavor, where it started from strengths in plant and microbial chemistry. It may evolve to be more active in other areas of science.
Such as like medicine?
Like medicine, yes, absolutely.
All right. And then you retired in December? Is that
Yes,
correct?
that's right.
How are you enjoying it, the retirement?
Well, I haven't really had time to enjoy it. So I retired my faculty line, but I immediately came back on board at a modified service position. So I'm not a faculty member per se, but I still have research projects that I'm engaged in. I still have people working for me in the laboratory on projects. I still collaborate with researchers in BDI, and I probably will do that until these projects that are funded come to a close. Yeah.
And then you also to take the time today to do this podcast.
Well, of course.
Yeah. What excites you the most about your field and your specialty? Of course, you can't pick a favorite, but the best parts, the multiple.
The best parts of my job have been to work with the many people that I have been able to work with. both the folks that have trained in my laboratory and have gone off to have successful careers of their own, as well as good friends who are scientists around the country and around the world who I have had the opportunity to interact with professionally and personally. So the interactions with others has been the most fulfilling and rewarding.
So it's just a community that you found.
Community.
That's awesome. And when it comes to, I did mention the future and what BDI can become and grow, what obstacles do you think may happen that could be overcome?
Well, there is always the struggle for funding. Having enough resources for the Institute to advance is always going to be a challenge. So that will be a challenge, but that can be overcome with the talented people that we have. I think that it's always challenging to keep researchers engaged with the Institute when they're pulled in many directions. All of us are expected to teach in our respective departments. We have demands in our respective departments, committee assignments, and things like that. So one potential obstacle is, how do you keep people engaged in the Institute and keeping the Institute forefront in their minds and their actions?
Yeah, I see. Mm-hmm. And when looking back into the past, I did look at your publications on ResearchGate. You have a huge amount, 256 is what I saw. And some of them date back to like about the time I was born.
Yes,
like 2004, 2005. Well, I was born 2003, but
okay.
You know. What was it like? Do you remember what it was like publishing your first paper?
Oh, absolutely. My first paper was published in 1987.
Oh, yeah. Okay. That's
so a few years before you were born.
A few years.
Yes, quite a few. Yes, I do remember that. I was a PhD student, and I was extremely proud and glad to be involved in that publication. It all kinds of things come to my mind when I think about that. We had to prepare our own figures for publication, and that involved a lot of photography for me at that time. And so, I learned a whole discipline of photography in having to prepare figures for publication. I thought that I was a good writer when I finished my undergrad and started my grad studies, but I recognized pretty quickly that learning to write essays in English is not the same as writing a good, clear, unambiguous paper in science. And so I learned a lot about writing in my first paper. I learned a lot about preparing figures, and then the just the conversion of data from your lab notebook to figures and tables in a publication really had an impression on me. It might be notebooks full of data that end up as a 5-page printed paper or 10-page printed paper. So many experiments that don't make it into the manuscript but still are required for you to make your conclusions. So I learned a real appreciation for the amount of work that went into publishing a scientific paper.
It's awesome. I also understand that you are the president of the American Society of Plant Biologists.
I am currently. I've been a member of that society since I was a graduate student, since 1987. Wow. That was the first place I went to a scientific meeting. It was the first place I presented my research at a scientific meeting. I met colleagues and my professional network and grew up within that professional society. That society actually publishes 3 journals. The first paper that I published was published in a journal published by the American Society for Plant Biologists. It was actually the American Society of Plant Physiologists at the time. They changed their name as things evolved.
In 2025, you did earn a prestigious title with the American Association for the Advancement of Science, or AAAS.
That's correct.
Is it AAS or triple AS?
People sometimes refer to it as AAAS. It is also a professional society. It's not focused on plants the way the American Society of Plant Biologists is. It's focused more generally on scientists. And so I belong to both of those societies. And AAAS includes social sciences, chemists, physicists, all sorts of scientists are members of the AAAS. So it's a much larger professional network that includes plant biologists as well. Yeah.
Want to tell me more about that one? The other science conference?
Yes, I try to attend. I really enjoy going to scientific meetings. I've been an editor of a couple of journals, and I get ideas for exciting research that I'd like to have published in our journals. By going to meetings, I also present our own work at meetings and enjoy doing that. And so I just really enjoy the in-person exchanges that I get when I go to professional conferences. So I probably go to 4 or so meetings a year. Often they're in the summer, occasionally not in the summer, but mostly are in the summer. ASPB, for example, they hold an annual meeting of their members, and people come and talk about plant biology in general. I'm also a member of the lipid research community as a lipid scientist.
Yeah.
They have a small community of lipid researchers, and they have a meeting every year as well, either in an international or a national venue.
All right. Well, Kent, thank you again for your time.
Thank you for your time.
Thank you.
Big thanks to Jacob for bringing us that interview. If you'd like to hear more from Dr. Chapman, who also happens to be the current president of the American Society of Plant Biologists, he will be the keynote speaker for BDI Research Expo 2026 next month. This year's expo will also include a celebration of BDI's 10th anniversary. Check the link in the show notes to learn more about the May 12th event. Remember, you can help us grow by telling a friend about the show, sharing it on social media, or leaving us a 5-star rating and review wherever you listen. You can also let us know what you think about the show directly by emailing podcast@unt.edu or calling 940-565-4341. Until next time, we hope you have a happy Friday, North Texas, and go Mean Green. Happy Friday, North Texas is a production of the University of North Texas. Today's show was produced and edited by Scott Brown with original reporting by Jacob Parker. For more information, visit unt.edu/podcast.